Welcome everyone. Again, my name is Tamas Shahida from CarboHyde, and today we'll talk about small molecule delivery with the help of cyclodextrins. This is the eighth masterclass of CarboHyde on cyclodextrins. We'll divide it, or I have divided the presentation into four main sections. First, we'll have a general overview about what cyclodextrins are, how they are used, what is the mechanism of action. Then I try to bring a lot of marketed products in late stages of development, examples of uses so that you understand the wide variety of applications there exists with the cyclodextrins. And so these will be real world examples and mainstream applications. And then I'll share a few new trends, new ideas, new developments, which are close to the marketed applications, but still have a twist, a hint of innovation that gives them some superior feature. And at the end, I'll share a few new cyclodextrins. So something that's again a little bit longer distance beyond what's available today, but what's coming in the future. So let's talk about generally of cyclodextrins. Many of you may know these molecules. So they have this donut or truncated cone shape that you can see here on the picture. And the core part is the cavity that you can see here or here. So the cavity of the cyclodextrin is somewhat hydrophobic, whereas the surface or the external part is hydrophilic, which means that they're very well soluble in water, but in the cavity they can encapsulate hydrophobic small molecules temporarily. So it's not a permanent interaction, it's secondary interactions. And as long as the guest compound is in the cavity of the cyclodextrin is somehow masking the presence of this molecule. So this means that it becomes water soluble, it becomes stable, it can mask the taste, the odor, and a lot of other features. And this is used in a myriad of products. Today we'll focus on pharmaceuticals, but we have other master classes for focusing on non-pharmaceutical applications too. The dominant user, by the way, is cosmetics, but there are a lot of food products, nutraceuticals, households, chemicals, and of course, pharmacy. Uh, what I mentioned is the main application, so this kind of excipient application. In the pharma field, there are upcoming uses, like uses as active ingredients, formulation of biologics that we'll not talk about today, stabilizing proteins, delivering genes, targeted therapies, diagnostics, theranostics, or uses in vaccines. That's the topic of other events. So what happens? How does this complexation happen? It's a thermodynamic interaction. The active ingredient doesn't really prefer the water because it's hydrophobic, but it prefers the cavity of the cyclodextrin, so it wants to enter. And once it's entering the cyclodextrin, it's a nice and cozy environment, which it likes. And we are taking advantage of this complexation. As I said, we can reduce the taste, the smell, or even interactions and side effects. We can lower the dose in some cases because the cyclodextrins, by helping solubilization, they can also help in improving the bioavailability and stability. But about these, we'll go into details soon. We try to make this interactive, so I have just, I think, two or three questions during. So type it in the chat box. What is the only administration route you think where there are no products with cyclodextrins currently on the market? In pharmaceuticals, of course. We don't wait long, so we'll type quickly. Inhalation products. Okay, that was cheating that came internally. But yeah, of course, at least our colleagues got it right. It's inhalation. Uh, so pulmonary application. So the cyclodextrins are very versatile in their use, so they can be loaded into any kind of drug formulation. So we're not just talking about oral solids. We can talk about sprays, liquids, suppositories, gels, any kind of semi-solids, creams, so really any kind of formulation. And accordingly, most of the administration routes have some products where cyclodextrins are among the components. When I mention cyclodextrins, I'm not talking about one molecule because cyclodextrins come in three different sizes. So the alpha, beta, gamma cyclodextrin. In this order, the size increases, and the chemical modification of some of them. As you can see, hydroxypropylation, where HB is mentioned, SBE is for sulfobutyl ether, beta cyclodextrin, and RM is randomly methylated cyclodextrin. So when talking about pharmaceutical uses, altogether, we're talking about seven different types of cyclodextrins that are used. Also, as you can see, not all of them is currently applied with all kinds of formulations, depending on their safety profile. Mainly, the regulatory approval varies. So, what does this look like? As I mentioned, cyclodextrins have different sizes of cavity. The alpha cavity is usually, or generally, as a rule of thumb, is a has a size that fits very well a linear aliphatic chain. The beta cavity is from a gas molecule's perspective the most versatile because that's the size of an aromatic ring or a small ring conjugation. So that where of course the majority of the pharmaceuticals fall. And then we have the gamma cavity, which is really for a bit larger molecules. The stoichiometry of the complexation is usually 1 to 1, but we have seen other cases too. So when it comes to the complexation itself, it would sound trivial that if we add more cyclodextrin, if we increase the presence of cyclodextrin, the drug loading, the concentration of the drug that we can bring into solution would increase linearly. And this is true in some cases, but not always. So as you can see, this A type of curves are close to linear, and depending on usually the stoichiometry, they deviate. This stoichiometry, they deviate from the linear, but are close to it. And usually these are the curves for the derivatized cyclodextrins. But there are some complexes that are not soluble in water at all, or just to some extent. And these kind of complexes have a plateau or complete insolubility in water. Considering binding strength, we can maybe distinguish three categories. So, of course, cyclodextrins do not complex anything or everything, do not complex everything. Some complexes are very weak. The majority fall into this moderate range, and some are really strong in the binding. What kind of techniques we can use if we want to make a cyclodextrin complex? Luckily, there is a little limitation there. So cyclodextrin complexes can be prepared in liquid phase, where you bring into solution the active ingredient, the cyclodextrin, where the complex is created, as I said, thermodynamically, and you can use different kind of techniques to isolate this complex in a solid form. So a cyclodextrin complex is usually a solid. Spray drying, lyophilization, and evaporation, precipitation, and so on. You can use semi-solid techniques, so this can be done in a kneading or an extrusion kind of way, and even it can be done in a solid phase with a milling technique. So this slide kind of summarizes the different type of concept. Usually what we do, that's the mainstream technique, we create a cyclodextrin solution, add the active ingredient, and obtain a vigorous stirring until the complexes are formed, which are then isolated. Uh, and similarly to this thermodynamic force, external strength in the case of the kneading method or extrusion can be used to force, we could say, the hydrophobic drugs into the cavity of the cyclodextrins. Why is it important to make sure that the cyclodextrin is complex? Is because one can think that, okay, it's enough if I just use the cyclodextrin, mix it with the active ingredient, and the complex will form in solid state inherently. This is not true. So what you will see here is that naked curcumin physically mixed with cyclodextrin. Here there is no complex. We just mix the two together, and here we actually form a complex. So let's see, especially the second and the third one. You can observe that, okay, it's still better than curcumin. The wettability is increased, even the solubility increased, but it's nowhere comparable to when a real complex is formed. So I guess the difference is rather dramatic and trivial. So making sure that the complex is formed is critical. Um, Also, another example to show you that if we obtain a physical mixture, this is remdesivir with sulfabutyl cyclodextrin, it's still not soluble in water, whereas a complex lyophilized or electrospun dissolves very beautifully in a short time. So, the it's nice that there are cyclodextrins that we can use to solubilize our active ingredients. There are quite a few methods and quite a few components that we can use to boost the performance of cyclodextrins. So these components help to stabilize the complex and to exist for a longer run, and thereby a higher portion of the active ingredient can brought to complex. So can you think of such ingredients that you think might work very well synergistically, we could say, with cyclodextrins? Okay, we have shy audience today. Anyhow, there are quite a few different approaches. So usually other solubilizers work pretty well in that case. So different types of polymers, different types of hydroxy and amino acids, and certain type of metals can help in that. Of course, if we just have the binary system of the cyclodextrin and the active, there are quite a few factors we can play with: the temperature, the pH, the type of buffer, and so on. But evaluating the performance, oh, sorry, I have to. Evaluating the performance of these third components is usually very economic because you can use less cyclodextrin and very useful. Just to bring you an example from a real world product, dissolving 10 milligram of domperidone. If you just use water, you need 2 liters. If you use a cyclodextrin solution, 62 milliliters. If you use, I think it was citric acid, 12 milliliters. But if you combine the cyclodextrin with a citric acid, you can go below 1 milliliter to dissolve the same amount. So that speaks for itself for the synergy. Another interesting part is the salt form of the active ingredient. Of course, the different salt forms of the active ingredients have different intrinsic solubility themselves, but not only that, it's also the when we add the cyclodextrins, you can observe that the improvement we can achieve with the same cyclodextrin is very different. So here you can see just an example: the hydrochloride salt from 80 microgram we brought to 2.4 milligrams. The tartarate had double solubility, more or less, as the hydrochloride, but here there is a five-fold solubility difference already, or with the sulfabutyl there is already a six-fold solubility improvement. So it does matter what kind of salt form do you use and do you pick for the active ingredient. Similarly for this ternary effect, especially if the counter ion is an organic, because with inorganic anions, cyclodextrins do not really interfere, but with organic, obviously, with organic ions, they can interact. So how do we evaluate if we could create this nice complex? Luckily, we have a myriad of techniques that are possible. So theoretically, what we say, not theoretically, practically, what we say that any analytical technique that can distinguish between a complex active ingredient and the naked active ingredient is suitable to detect the complexation. So I don't know, just an example, the elution time on an HPLC of the naked active ingredient would be different than a complex active ingredient. It will interact differently with the column. Same for most of the chromatography techniques. The chemical shift in the NMR will be different. The UV absorption will be different, and so on. So these are all potential tools to detect it. Of course, the most straightforward, so these are more or less indirect confirmations. Direct confirmations usually come from solid state methods like DSC or X-ray, where we can see the crystallinity disappearing. Um, or the changes in thermograms, or even, of course, with a microscope detecting the complex itself. What else do we use to analyze the complexes? We can assess the binding strength with all these techniques that we mentioned. So these are all suitable for a kind of titration and assess the binding strength of the interaction. What do you think, in your opinion, how good is the correlation between the molecular stoichiometry, what I showed, that usually it's one active ingredient fitting into one cyclodextrin, and the actual stoichiometry that we use in the product? Do you think it's in a good correlation, or that correlation is not as good? Okay. Amandsen, it's not good, and you're completely right. It's not good because actually there is almost no correlation between the molecular stoichiometry and the real world stoichiometry, what it's in the product, because how much cyclodextrin we had to add in the product, how much cyclodextrin excess we have to add to the product is much more dependent on the binding strength. How good is the fitting of the active ingredient into the cyclodextrin, not the actual stoichiometry or the ratio. But still, we can determine this ratio. There are all kinds of complexes, and usually it's the job plots technique that's a unique titration that's used for this one. What's more interesting is the geometry of the complex. So how the active ingredient is fitting into the cavity of the cyclodextrin, which rim is it entering from. So as you can see, the two rims are not identical. One is wider, one is narrower. This kind of considerations are usually very useful to explain how the cyclodextrin complex is formed and how it dissociates. And of course, like one of our guests said, we can use molecular docking to prove the same. And of course, once the complex is done, less analytical and more uh, applicational analytics are used. So how the active ingredient dissolves from the cyclodextrin cavity, what are the kinetics, how different is the diffusion through membranes, protein binding, and such. So these in vitro tests are very common too. So let's see a few marketed products and examples of uses of cyclodextrin. So as we said, typical what we are seeing solubility improvement. I would say that even today it's the mainstream. 90% of all the uses of cyclodextrins are to improve the solubility of some um, insoluble drug. Hexalone, very nice product can be created. Of course, the yeah, and as an advantage, these products can be, of course, parenterals, oral solutions, liquids, whatever. There is another application. I'll explain how it works because it's a bit tricky, but you can also use cyclodextrins to enhance the permeation of certain products. This is the example of Baqsimi. It's an Eli Lilly product, and it's a nasal product, as you can say. And in this case, the cyclodextrin is used dominantly to improve the bioavailability of the product, increasing the stability, usually the shelf life, not the in vivo stability of products, is also something that's used a lot of times. The big advantage of using cyclodextrins is that you achieve all three with the exact same compound. So if you form a good cyclodextrin complex, you will see the solubility improvement. You will see probably the stability improvement. I explained why I said probably. And you can also experience the permeation improvement at the same time. Bioavailability enhancement, quite remarkable. As you can see, it's an example of coenzyme Q10 almost 20 times, making oils into powders, or that's also improving bioavailability of or absorption of certain compounds very dramatically. When it comes to stabilization, cyclodextrin can protect compounds, or at least are reported or known to protect compounds from almost all kinds of external forces, so light, heat, oxygen, pH. The most tricky is the moisture. And that's because usually cyclodextrin complexes are formed in aqueous environment. That's how cyclodextrins can be solubilized, and that's the easiest way for active ingredients to get into the cavity of the cyclodextrin. Once it's in the cavity of the cyclodextrin in a solid form, the cyclodextrin can protect it from the external forces, or the moisture, the water. But of course, it has to survive. The active ingredient has to survive until it gets into the cyclodextrin cavity. But there are non-aqueous complexation methods for that too. So enhancing permeation is possible with cyclodextrins, but it's not always working, because in order to enhance permeation of active ingredients, there has to be a high local concentration of the cyclodextrin. So if you swallow a tablet with a complex within, and that dissolves in 2 liters of gastric media on a huge surface of the GI tract, the local cyclodextrin concentration at a given fraction of the GI tract will be very low, and it will not mean a permeation improvement. But if you apply a nasal spray with a cyclodextrin complex, or a topical cream, and that's why it's one of the reasons why it's very popular in cosmetics, that's a fairly high local concentration of the cyclodextrin. And the cyclodextrin in this case cannot only solubilize, or will not only solubilize the active ingredient, but it will also solubilize certain components of the membrane, like how you can see here, the cholesterol, the fatty acids, giving the membrane a fluidity and improving the permeation of certain of the active ingredients. So that's how permeation enhancing works, and also the limitations. So what is there beyond solubilization? Taste masking, as I mentioned, usually for compounds that come at low doses. Content uniformity, some hormonal compounds are formulated with cyclodextrins for that. Fast dispersion and dissolution. Cyclodextrins are really ideal for ODT type of products, because the complex itself, upon dilution, so how the active ingredient is released from the cyclodextrin cavity, again, thinking about the thermodynamics, it's upon dilution. So when the complex gets dissolved and there is competition outside, or there is a dilution factor, the active ingredient gets released. And that can happen in the mouse as an ODT gets dissolved, and then a huge dose of the active ingredient can be released, even supported by the permeation enhancing effect. It's a very nice application. Chemical stabilization, this example is very interesting, so I'll tell later. And life cycle management. So, okay, fast onset. Now it's easy to understand why it's a fast onset, because of the very quick release from the cyclodextrin cavity, but life cycle management is also there. I will not go into details here. I just wanted to show you how versatile cyclodextrins are when it comes to designing different types of formulations. You can see there are gums also in the nutraceutic space. There are a lot of cyclodextrin containing gums, gels, lotions, whatever, even wipes. So yeah. So there are a few advanced applications that we can talk about with cyclodextrins, so that are a bit more complicated. Uh, Developing super generics is one of these with cyclodextrin. So uh, cyclodextrin, as you could understand, cyclodextrins improve the solubility of the compounds. They improve the bioavailability of the compounds. So now it's easy to understand that if we compare, especially in talking about oral solids, a cyclodextrin complex administered orally to a non-cyclodextrin complex administered orally, usually they won't be bioequivalent because the cyclodextrin influences the onset, it influences the total blood levels, the kinetics, everything. But developing super generics, so repurposing drugs with cyclodextrins, is something that's very popular. Like I said, they can help in complicated formulation challenges like chemical stabilization, permeation improvement, even reducing osmolarity. I'll bring you an example of that. One particular application is fairly difficult with cyclodextrins. So now you understand that inherently they have a tendency to release the active ingredient very fast. So what if I don't want the active ingredient to release very fast? I want a controlled or an extended release. In these cases, you have to incorporate cyclodextrins into other drug delivery systems or the complexes into other drug delivery systems. I'll show a few examples of those. And targeted delivery, that will be the last part that we'll tackle. So developing generics with cyclodextrins usually happens for liquids, and even with liquids, it typically happens with parenterals because in that case, the fast release is not altering, of course, the blood levels. But we have also seen such in ophthalmics. And in this case, usually the strategy is that cyclodextrins are used to replace old, outdated excipients of known formulations. So that's how the IP is generated. That we replace polysorbates, organic solvents, preservatives, whatever, with cyclodextrins, and have a safer formulation at the end of the day. Stabilization with cyclodextrins is a bit more difficult topic because when I want to solubilize something with cyclodextrins, if I can make a decent complex, I'm good. It will improve the solubility to some extent anyway. I'm not saying that it will be a fantastic improvement, but if the complex is formed, solubility is improved. That's guaranteed. When it comes to stabilization, this is not enough because you have to incorporate the sensitive part of the molecule into the cavity of the cyclodextrin in order to achieve the good stabilization. So this product is very particular or peculiar because it contains two different cyclodextrins. It contains both the alpha and the beta cyclodextrin, and as you can say, they complex different parts of the molecule. And what does this mean? When we see the impurity formation with the beta cyclodextrin, here we can see the tendency with the alpha cyclodextrin, and when we combine the two, we can see that that's really where the magic happens and where the stabilization, the complete stabilization is achieved. So this kind of fine-tuning on the complexation, where when the chemical stabilization is the goal, is critical. So let's see how we can achieve these innovative applications with cyclodextrins. Just a few examples, it will not be comprehensive. So there is a technology called OptiReach, which enables delivering active ingredients to the back of the eye with cyclodextrins. And it's also very interesting because it exploits aggregation of cyclodextrins, which is supposed to be a not very nice thing when it comes to drug formulation. So this works only with gamma cyclodextrins, at least as far as we currently know. The complex is created and the complexes aggregate, forming these microparticles. And once administered to the eyes, these microparticles can penetrate into the back of the eye and replace ideally intravitreal injections. And this technology is so successful that the first product will be marketed, I think, still this year or at the beginning of next year. But several phase 3 studies have already been uh, tackled by Oculis for the first active, and there are more actives to come. So it's a wonderful technology. What if we want to achieve, again, a targeted or an extended controlled release? Just a few examples. This is not something that's on the market or close to the market, but we can incorporate cyclodextrin complexes into liposomes or other lipid nanoparticle systems fairly easily. That would be a nice solution. Or we can combine the cyclodextrins with different type of polymers or other matrices where the degradation of the matrix is the limiting factor. And of course, as soon as the complex is available to get in contact with the environment or the media, the active will be released, but it will be still an extended release. This is something where we have substantial experience and a lot of data that you can achieve wonderful extended release with the cyclodextrin. So here the cyclodextrin would be the solubilizer, and another component would play the role of controlling the release itself. So how can we achieve reducing osmolarity? Two things. Cyclodextrins themselves do not contribute, okay, except for the sulfabutyl ether cyclodextrin, which is a polyanion, uh, do not contribute that much to osmolarity. So you can use a fairly high concentration of the cyclodextrin and still be within the isosmotic range. The problem was with this particular cyclodextrin, the sulfabutyl ether beta cyclodextrin, which is a polyanion, as you can see here. These sulfabutyl groups with the sodium counter ion. So 10% of this cyclodextrin was already isosmotics. And this company called Baxon figured out a way that, okay, they said that we will not use the cyclodextrin cavity, we replace the sodium ions and use the active ingredient as the counter ion. Of course, it's not working with everything, but with some compounds, it's working beautifully. And the other, okay, it's not just that it doesn't it doesn't add to the osmolarity, but also, as you can see here, the ratio between the active ingredient and the cyclodextrin completely changes. Earlier, we were talking about 1 to 1 or close to that. Here, as you can see, we're talking about 6 actives to 1 cyclodextrin. So in very small volumes, you can concentrate active ingredients with this technology. Again, not working with everything, but where it's working, it's beautiful. Microneedles, it's also a coming application. I was really fond of this technology. I think that, okay, cyclodextrins have a huge potential there, but I also thought that, okay, this is maybe a bit more science than real world. And then recently, I met a company who has a microneedle technology already available on the market, although it's a cosmetic application, but still. Cyclodextrins are suitable to build microneedles themselves, or you can use the complexes without any other component, or you can combine with others, but it's a very nice fit with cyclodextrins. There are a few initiatives to use modified cyclodextrins to counteract the drawbacks that the marketed derivatives cannot achieve. So as we said, okay, sustained or controlled release is not available or not a possibility with current cyclodextrins. we can create cyclodextrins that can have a sustained or controlled release. And these cyclodextrins would be the ones that are not soluble in water. So you can create cyclodextrins modified with alkyl chains like ethylated or acylated cyclodextrins. If you use the complexes of such products, then the then the complex would release the active ingredient slowly. Another possibility to achieve that would be to immobilize the cyclodextrin to some surface, and this is something that they have also tried in human studies, so like hernia mesh and or the inside of catheters and stuff like that. So you can use either polymerized cyclodextrins or cyclodextrin reactive cyclodextrin derivatives to attach them to the surface of certain medical devices, load them with the active ingredient, and in this case, the release of the active ingredient will be more gradual than with the water soluble versions. And talking about cell and organ targeting, here we are definitely need modified cyclodextrins. So the chemistry of the cyclodextrins is very versatile and enables us to create a lot of wonderful derivatives. So for instance, we can tag them with folic acid that can target different type of cancer cells, or we can tag them with sugar molecules like mannose and lactose. And as you can see, such tagged cyclodextrins can target and accumulate in certain parts of the body. So, uh, through sugar receptors, lactose appended cyclodextrins can enter the brain, or they can accumulate or yeah, accumulate in the liver, kidney, where technically you can target any kind of organ or cell type if you have a good receptor. By themselves, by the way, parenterally administered cyclodextrins are excreted via the kidney. So that would be the basic organ where cyclodextrins would excrete, and where you could detect them at higher concentrations. If you want other organs to be targeted, you need some modification. So that's what I had for today's talk. I hope I could provide you some insight into the uh, large variety of applications of cyclodextrins to deliver small molecules, what is possible today, what is happening today, and what may be the new possibilities for tomorrow. So I really hope it was easy to follow and enjoyable. And now, if you have any questions, feel free to drop them in the chat box. And oh, that's my daughter. And I'm happy to answer. Thank you. I'll stop the sharing and I'll open the. Okay. So, one question, does the active ingredient accommodate inside the cyclodextrins in these systems? So I don't know when you dropped the question, what kind of systems are we talking about, and what do you mean by accommodating? If we're talking about so when you, uh, for instance, the liposomal or combining the cyclodextrins with a polymeric system, then the answer is yes. In those cases, uh, yeah. So with the polymers, yeah, in those cases, technically the cyclodextrin complex itself, so the active ingredient loaded into the cyclodextrin is incorporated into the matrix and in the complex form. So yes, the short answer is yes. That's how those are happening. NMR on those. NMR to determine what? That the active ingredient is still within the cyclodextrin cavity? I don't think so. I'm not exactly sure that. Ah, okay. To determine the structure in those cases. I don't recall. So in those cases, usually when you already incorporate the cyclodextrin complex into another drug delivery system, typically it's functional studies that we are uh, doing. When it's only the cyclodextrin and the active ingredient, we yeah. That would be very tricky in that case too. So you mean to justify that after we loaded the complex into a polymer system, the cyclodextrin complex is still intact and it's still existing? No, I don't. I'm not aware of any studies like that. It could be very tricky. Maybe you're up to the challenge. I don't know. It's not easy to do, and I don't remember anyone going for such a challenge. But of course, I don't know all the literature. It's an interesting idea. Please feel free to ask anything that's on your mind. What is the mechanism? How cyclodextrin is crossing the blood-brain barrier to deliver the loaded small molecules to brain. So, cyclodextrins. So here we're talking about two different concepts. The cyclodextrins that are commercially available cannot really cross well biological barriers. So the bioavailability of cyclodextrins, if administered orally, is below 5%. Usually. And that is true for, well, even more true for the blood-brain barrier. So I think it's the highest values are around 2% for the cyclodextrin. So when we just administer a cyclodextrin complex with the hope of delivering more active ingredient into the brain without any targeting, what is happening that in reality we only make more active ingredient available in the circulation, but we are not supporting the permeation at all, or the penetration at all. So cyclodextrin only goes to the brain through passive transport and also their very small ratios. And if we're talking about the complex will not exist. So what you can imagine, we have a complex, we inject the complex, there is a huge dilution, so the complex falls apart immediately. The cyclodextrin circulates and 2% goes to the brain, and the active ingredient circulates attached to different proteins, whatever, uh, and it can reach the brain at a higher dose. This is what would happen with a typical complex. If we tag the cyclodextrin with something that can find a receptor on the blood-brain barrier, like the lactose we said, or certain type of peptides, or use cyclodextrin polymers that are bigger molecules, then we can aim for active transport, and that's why there is a higher ratio of the brain or an accumulation in the brain that we detected. That's because in that case we are triggering active transport. So that's how the cyclodextrin gets into the brain. Here the challenge is, and this was confirmed in vitro, but also in some in vivo experiments. So the cyclodextrin can go to the blood-brain barrier and it can get inside via active transport, but can it carry the active ingredient with it? So for that, you need a strong complex, a nice complex, so that it doesn't fall apart immediately. And that's realistically, that's a very challenging task, something we work on, but it's difficult to manage that. Yeah, welcome. If there are any questions, let me know. Yeah, I really appreciate the compliments. It's heartwarming that you like the presentation. I hope it was useful. In any case, if there is no further questions, maybe something will pop your mind later. You can reach out to us on any platform that you wish. Thank you for joining today. It was a pleasure to have the masterclass, and we'll continue with other topics in the future. And you all have a very nice afternoon, nice day, depending on where you are. Take care. Bye bye.